Thank you all for coming to my talk today. This afternoon I'll be discussing the use of environmental DNA as an effective tool for tracking the dispersal of larval fish in Lake Michigan. This summer I was advised by CIGLR's Chaganti and NOAA GLERL's at Rutherford. So first, a little bit about me. I'm an undergraduate student at the University of Minnesota, and I am pursuing a major in fisheries and aquatic science. I'm also pursuing minors in genetics and computational biology. So before we dive into the really awesome genetics and physical modeling behind this project, it's important we understand the what and the why. What are larval fish? Why are they important, and why should you care about them? So the term larval fish describes a developmental stage in a fish's life where their fins are not fully developed and they're within about the first 40 days of hatching. Additionally, they're only a few millimeters long, so they're incredibly difficult to identify and have very limited swimming abilities. Broadly speaking, their survival directly influences the abundance and distribution of adult fish. So Lake Michigan specifically is home to invaluable living natural resources that are at the center of the Midwest recreational fishing economy. In particular, yellow perch and alewife are crucial points in Lake Michigan's food web as they serve as primary food sources for salmon and other predatory fish, which are central to the Lake Michigan fishery. As larvae, yellow perch and alewife are subject to various challenges during their early life stages. Both alewife and yellow perch spawn in nearshore environments, which makes their larvae highly susceptible to advection via lake currents and circulation patterns due to their limited swimming ability. In combination with the introduction of invasive species, as well as overfishing, the transport of these larvae into unsuitable habitats when they are only a few days old could explain the recent declines in that both yellow perch and alewife stocks have experienced. Ultimately, tracking the advection of these fish can provide great insight into the recruitment success and survival of nearshore spawning fish species. So to catch these larvae, the NOAA and CIGLR team uses both bongo nets and Newston nets. The bongo nets are pictured on the far left and sample the whole water column, while the Newston nets are pictured in the middle and sample just the surface. Additionally, we use a hydrodynamic numerical model to predict where these larvae are transported based on previous catch data and currents. The model output for each day of our cruises informs where we sample for larvae. However, I'll talk more about that later. So these methods are far from perfect. Our netting surveys can easily miss larvae throughout the tow and have some selectivity bias. Additionally, it is often a labor-intensive process that involves a crew of scientists going out on a research vessel for days at a time to track the movement of these fish. So a new approach that was the focus of my summer here at CIGLR is called eDNA, which is shorthand for environmental DNA. We're interested in whether or not the use of molecular techniques can provide accurate insights into the advection and distribution of larval fish. More specifically, can we use eDNA to determine if a certain organism is present and if it is effective for relative abundance estimates? Thus far, lab experiments show promising results. However, it is a lot less clear how field studies will hold up. So for our eDNA workflow, we begin by collecting water at any given depth using what is called a Niskin bottle. At each site, during the study, we collected water at the surface and at the bottom of the epilimnion. While we would then immediately filter the water on the boat from both. Well, we'd immediately filter the water on the boat and then preserve those filters in liquid nitrogen to preserve the DNA. Then back at the lab in Ann Arbor, I spent day and night extracting DNA from these filters. This is where I primarily allocated my time this summer. And yes, this video is in slow motion. This is just how fast I work. Next, we would use the qPCR machine to get an estimate at the initial concentration of target DNA in that sample to then determine whether or not a certain species is present and estimate the relative abundance. So as I stated in the previous slide, qPCR is what we use to quantify DNA concentration in each sample. qPCR stands for quantitative polymerase chain reaction. These reactions are based on a thermal cycling regime that amplifies a certain target piece of DNA exponentially and then quantifies that DNA with fluorescence. The number of cycles it takes to reach a fluorescence threshold can then be used to quantify and compare the initial concentration of DNA in and between individual samples. We use species-specific primers for yellow perch and alewife such that only DNA from those organisms was amplified. And ultimately, a lower CT value indicates a higher initial concentration of target DNA. And finally, before we dive into the results, it's critical to understand our sampling regime, which is entirely informed by this hydrodynamic model. So the Lake Michigan and Huron Operational Forecast System, aka LMHOFS, is what the physical modelers here at GLERL use to track the dispersal of larval fish. On the first day of each research cruise, we would sample multiple transects, and when we caught fish, we would call up our physical modelers with the coordinates of these fish. They would then simulate the release of 2,000 particles in their model and then track the particle density throughout the cruise. Researchers here at GLERL have aptly coined the model output as the blob, and the X in the middle of the blob is the centroid and represents the point to have the highest particle density predicted by the model. We would sample the centroid and the 0.1 density section as well as outside of the blob. So getting into our initial results, I spent the first half of the summer extracting and processing DNA from samples collected by one of last year's fellows. It is important to note that for both July 2023 and July 2024, no yellow perch were caught and there was no eDNA signal from yellow perch. So all the data presented from here on out represents just alewife. So here we have the surface samples from July of 2023, and the map on top shows the number of fish caught at each site. So the larger the orange circle, the more fish were caught at that site. And then the map on the bottom represents our eDNA signal with pink circles. And like I said earlier, CT value is inversely related with target DNA concentration. So bigger circles have a lower CT value. One quick important note here, the unfilled pink circles do not indicate the absence of alewife DNA. They just suggest that the CT value at this site is simply above 35, indicating that the initial concentration of target DNA is too low to reliably quantify. And upon initial glance, there's really low correspondence between the model output and the fish caught. However, this is actually a really exciting result. The absence of larval fish on the surface within and around the centroid is consistent with the widespread observation of daily vertical migration of zooplankton. So during the day, zooplankton remain closer to the bottom to avoid predators and migrate to the surface at night. Now moving to the bottom samples, we immediately see much higher fish catches and eDNA signals and general correspondence to our model output, as it appears the larval fish follow the zooplankton behavior of DVM. Additionally, we see that distance from shore appears to be a confounding predictor of larval fish. This makes sense as the model tracks only advection of fish caught at the transects near the top of the map. And we are likely picking up fish and eDNA signal from alewife larvae that had not been advected from shore. For the second half of the summer, I had the opportunity to participate in year 2's research cruise in early July. We sampled M15 and M45 transects on day 1 and used the Lake Michigan Huron Operational Forecast System to inform our sampling for the next 4 days. Sites where there was a positive eDNA signal are green, while sites where there is no eDNA signal are red. Sites where larval alewife were caught are denoted by stars, and sites where no larval alewife were caught are represented by a circle. Here we have a total of 5 instances in which netting surveys and eDNA agreed, 3 true positives and 2 true negatives. Additionally, at 6 of the 11 sites, we detected the presence of alewife DNA, but not with netting surveys. Moving to the bottom samples, here we have 6 instances where both fish were caught and positive eDNA signal was detected. There were 5 instances where no fish were caught and a positive eDNA signal was detected. One really important result of this study was that out of all the samples from July 2023 and July of 2024, there were no instances where we caught fish but didn't detect the presence of alewife with eDNA. So now, how do our NetToes, eDNA methods, and hydrodynamic modeling methods correlate? The graph on the left represents the relationship between the model prediction and the eDNA signal. The x-axis are the sites ordered in increasing probability of alewife presence from left to right. And on the y-axis are the CT values, which, like I said earlier, are inversely related to eDNA strength. So overall, here we would expect to see a negative trend. The graph on the right shows the relationship between the model prediction and the number of fish caught at those sites. Here, we would expect to see a positive result. Generally, we have low, if any, correspondence between the model output and both the number of fish caught and the CT values. This is not surprising, however, because the model is tracking a certain group of fish that was initially sampled on day 1. And like I said earlier, this eDNA signal and fish catches that are outside the blob are likely larval alewife that had not yet been infected or were not simply in the initial group of fish sampled. Finally, here we have the number of fish caught versus the CT values. So here we would expect a negative correlation. There's a lot of error associated with our netting surveys. These error bars represent the standard deviation around the number of mean fish caught at each site. Additionally, since CT values above 35 are not considered reliable for accurate estimates of concentration of DNA, here's a new regression line omitting observations where the CT is above 35. Still a very weak negative correlation is present. So while in lab experiments, eDNA has been demonstrated to be useful, there are many uncontrollable variables in our field study which makes validating any of our methods really, really difficult. Obviously, netting surveys don't capture exact abundance and have some selectivity issues, and our hydrodynamic modeling model only tracks the fish captured on the first day of the cruise. For our specific study goals, eDNA has proven to be a very effective technology. We are not interested in understanding whether or not alewife are on the surface or on the bottom, but rather their dispersal via advective currents. As shown on previous maps, there were many sites where no fish were caught on the surface and many fish were caught beneath the surface. All the while, we had eDNA signals at both depths. So in conclusion, here we present a total of 70 samples across 3 cruises during the summers of 2023 and 2024. Our goal was to use traditional and novel methods to track the advection of 2 fish species of economical importance in Lake Michigan. Our 3 methods, ichthyoplankton netting, hydrodynamic modeling, and eDNA are complementary, while eDNA has especially shown to be an effective, cheaper, and accessible option for inferring presence and absence. In terms of relative abundance, eDNA in this case has some challenges due to the incredibly low density of larval fish in Lake Michigan. And some future directions for this technology is the development of a qPCR assay that targets transcripts only expressed in larvae. So our current assay does not discriminate between larval and adult alewife. However, this is not a main concern as adult alewife do not remain near shore after spawning, and we would not, generally speaking, expect to see adult alewife where we're sampling for larvae. Additionally, the use of eRNA concentrations could be used to calculate the age of eDNA, as eRNA and eDNA have different degradation rates. And with that, I'd like to give a very special thank you to my advisors for this summer, Chaganti and Ed Rutherford, and additional thanks to Lucas Vanderbilt and Tate Algayer, who are part of the CIGLR Molecular Biology Lab and played a very crucial role in my training in the protocols, to Maddie Tomczak, Paul Gleishaw, Eliza Lugton, and Captain Andrew Galia, who are all on the 2024 Larval Fish Cruise, and to my fellow fellows and interns here at GLERL. And of course, a huge thank you to Mary Ogdahl and Margaret Throckmorton for making this wonderful fellowship possible. with that, I would like to thank you all for watching my presentation, and I would be happy to take any questions.
Great job, Christian. And we've got time for a couple questions.
That was a great presentation. Congratulations, first of all.
Thank you.
I also had a question. When you first showed the graph early in the presentation about the fish populations with the yellow perch and the alewife, I'm not sure if I'm pronouncing it right. Alewife, thank you. I noticed a spike in the yellow perch population in 1990 that seemed pretty dramatic, and then a sudden drop off. And I wondered if you could talk about that a little bit and what might have happened and cause that drop off, spike and then fall.
I think in terms of yellow perch, I'm pretty sure that crash is related to overfishing. Yeah, I'm getting a nod from Ed. So yeah, that's where are we? Yes. Yeah. Yes, overfishing.
Okay, this is more of a personal question because we've talked about it before, but I know you enjoy doing eDNA work with fish specifically, but I guess and you mentioned to me on an occasion that you didn't see yourself completely going into eDNA, but in the future, do you think you could transfer what you learned here into like a fisheries position at all?
Absolutely, yeah. I mean, Rao and Ed, as my advisors, made sure to give me a very broad experience here. So I learned a lot of really, really awesome molecular techniques that are not specific to eDNA whatsoever, and also completely new experience for me going on the boat, towing these ichthyoplankton nets, and even seeing some really, really awesome ROV deployments and stuff like that. So.
Okay, congratulations on your presentation. It was really great. Here's a fun question. What was your favorite part of the cruise?
Definitely going on the we went out from noon to midnight on Thursday, and that was my favorite. Seeing the sunset and then also towing some nets at night. That was really cool. So
yeah. So do you have any theories on how eDNA is correlated to the number of fishes? Like where does eDNA come from the fish tissue, and how are they affected by the environment factors?
Yeah, absolutely. So so eDNA generally can be, it can be from sloughed off cells, it can be from fecal matter, it can be from, like, we'll see a huge jump in eDNA signal if we were to sample during the alewife spawning events. So with larval fish, yeah, it's going to be sloughed off cells. And sorry, what was the second part of your question? Yeah, there may be some implications for like depth, as our lakes are stratified, and generally eDNA is going to be more stable at colder temperatures. So maybe on the surface we might be getting lower signal because of the increased light and higher temperatures, and it might also translate towards the bottom where we're getting better signals. So.
Great. Thanks again, Christian.
Thank you.
Great job.